Bismillahirrahmanirrahim. Today I welcome all my students of second year MBBS to the almost you can say the introductory lecture regarding the GIT. Now, it is very important to learn about the neural control of gastrointestinal function. The GIT is having its own nervous system and that has been given the name of enteric nervous system. So what is it? What is it comprising of? What are its functions? What are its locations? We are going to learn about this in detail in order to learn how the GIT is functioning, how the secretions they are being controlled, and how it is all working. So, as I have told you that GIT is having its own nervous system, which has been given the name of enteric nervous system. So you see that where it is exactly lying is in the lining or in the wall of the gut, and it begins right from esophagus and extending all its way down to the anus. Now, it is so surprising that enteric nervous system is having around almost 100 million of neurons. What are neurons? Neurons are the structural and functional unit of nervous system. So around 100 million of the neurons, they are lying there in the enteric system. So, which is almost entirely or exactly the number of the neurons which are there in the spinal cord. So, see how important it is. So, this highly developed enteric nervous system is especially important in controlling the gastrointestinal movements and secretions. Now, the plexus of enteric nervous system. What is the plexus? You all know better than myself that the plexus is actually a sort of web or collection of different neurons or nerves. So, mainly it is composed of two plexuses. One is lying in the outer muscular layers, and the one is lying in the inner area, that is in the submucosal. So the outer plexus is lying between the longitudinal and circular muscle layers and are being given the name of Myenteric plexus or Bashes plexus. Whereas the inner one, which is lying there in the submucosa, has been given the name of the submucosal plexus or Meissner's plexus. Now, these two plexus, although they are lying in the different layers, but they do have connections with one another. And the myenteric plexus is going to control the gastrointestinal movements, and the submucosal plexus is controlling the gastrointestinal secretion and the local blood flow. Now, sympathetic and parasympathetic fibers, they are connected to both the myenteric and submucosal plexus, and stimulation of either of the parasympathetic or sympathetic can either greatly enhance or inhibit the gastrointestinal functions. Now look at this diagram. This is going to make you learn or make you help to know the different plexus, how where they are located. One is close to epithelium lying in the submucosa, given the name of submucosal plexus. The other one is close to the outer lining, that is between the muscle layers, that is the myenteric plexus, and they do have the connections with one another in between, and they do have the sympathetic supply as well as the parasympathetic supply, and how they are working we are going to learn in detail. Now, the sensory nerve endings, which originate in the gastrointestinal epithelium or gut wall and send efferent fibers to both plexus of enteric system, to the prevertebral ganglia of sympathetic nervous system, to the spinal cord, in the vagus nerves, all the way to the brain stem. These sensory nerves can elicit local reflexes within the gut wall itself and still other reflexes that are relayed to the gut from either the prevertebral ganglia or the basal regions of the brain. Now, it is very frequently asked that what are the differences between these plexuses, these plexuses. Now, one is the myenteric, my from muscles, submucosal, close to mucosa. So, one difference is that one is a linear chain of interconnecting neurons extending the entire length of gastrointestinal tract. Now, the other is controlling the function of the inner wall of each minute segment of the intestine. Myenteric Plexus is concerned with controlling the muscle activity along the length of the gut, whereas it is the many sensory signals originate from the gastrointestinal epithelium and are then integrated in the submucosal plexus. Increased tonic contraction or the tone of gut wall is its function, whereas the release of different secretion is the function of submucosal plexus. Some of the neurons are inhibitory, and how they are inhibitory because of the release of the inhibitory transmitter, possibly the VIP or some other inhibitory peptide, whereas increased intensity of the rhythmical contractions, slightly raised rate of rhythm of contraction, increased velocity of conduction of excitatory waves along the gut wall, causing more rapid movement of the gut peristaltic waves. Whereas the function of the submucosa is to control the local intestinal secretions, the local absorption, the local contraction, causing infoldings of the mucosa only. Now, the different neurotransmitters which are related with that of the enteric nervous system, they are acetylcholine, norepinephrine, adenosine triphosphate, serotonin, cholecystokinin, substance P, vasoactive intestinal polypeptide, somatostatin, leuenkephalin, metenkephalin, bombesin. See, from the different cells which are lying over there in the lining of the gut, they are releasing these different neurotransmitters. See, how the autonomic control of gastrointestinal tract. Autonomic control means the control or the function which is not under our will. We cannot control it. You all know that we are, although you are sitting, lying, standing, you don't know what is going on in your gut. So that is not within your own control. So that is autonomic control is by that of the parasympathetic and sympathetic nervous system. The parasympathetic is having the craniosacral outflow, whereas the sympathetic nervous system is thoracolumbar outflow. And about the autonomic control, we are going to learn about it in detail later on, along with the CNS. Um, Autonomic control regarding the parasympathetic function, you should all know that parasympathetic is going to increase. The main myth is actually you have to learn that rest and eat. That is the function of the parasympathetic. This is actually functioning when the person is at rest and is this process of digestion going on. So it is enhancing the gut motility as well as the secretion of the gut related to gut. So almost entirely the cranial parasympathetic nervous fibers, they are almost entirely in the vagus nerves, and these fibers they provide the extensive innervation to esophagus, stomach, pancreas, and somewhat less to the gut down through the first half of large intestine. Then we have the sacral divisions, which originate in the second, third, and fourth sacral segments, and they pass through the pelvic nerves to the distal half of large gut and all the way to the anus. So, the sigmoidal, rectal, and anal regions are considerably better supplied with parasympathetic fibers than are the other intestinal areas. These fibers function especially to execute the defecation reflexes. Stimulation of sacral parasympathetic nerves causes general increase in activity of entire enteric nervous system. Sympathetic stimulation usually inhibits the gastrointestinal tract. The sympathetic fibers to the GIT originate in the spinal cord between segments of T5 and L2. Most of the preganglionic fibers that innervate the gut after leaving the cord enter the sympathetic chains, which are lying on the either side of spinal cord, and many of these fibers then pass on through the chains to outlying ganglia such as to the celiac ganglion and various mesenteric ganglion. Postganglionic fibers then spread through postganglionic sympathetic nerves to all parts of the gut. The sympathetics innervate essentially all of the gut rather than being more extensive nearest the oral cavity and anus, as is true of the parasympathetics. The sympathetic nerve endings secrete mainly norepinephrine but also a small amount of epinephrine. Stimulation of sympathetic nervous system exerts its effects in two ways: to a slight extent by direct effect of secreted norepinephrine to inhibit intestinal tract smooth muscle, to a major extent by an inhibitory effect of norepinephrine on the neurons of enteric nervous system. Strong stimulation of sympathetic system can inhibit motor movements of the gut so greatly that this can literally block movement of food through the gut. Um, Afferent sensory fibers from the gut, um, they can be stimulated by the gut mucosal irritation, distension of the gut, presence of different chemical substances in the gut. Signals transmitted to the fibers can then cause excitation or, under other conditions, inhibition of intestinal movements or intestinal secretion. Other signals from the gut go all the way to multiple areas of spinal cord and even the brainstem. For example, 80% of the nerves fibers in the vagus nerves are afferent rather than efferent. These afferent fibers they transmit sensory signals from the gut into brain medulla, which in turn initiates vagal reflex signals that return to the gut to control many of its functions. The anatomical arrangement of the enteric nervous system and its connections with the sympathetic and parasympathetic systems support three types of gastrointestinal reflexes, which are essential to gut. They are following reflexes, which are integrated entirely within the gut wall enteric nervous system, and these include reflexes which control much secretion, peristalsis, mixing, local inhibitory effects, and so forth. Reflexes from the gut to prevertebral sympathetic ganglia and then back to gastrointestinal tract. These reflexes transmit signals long distances to the other areas of the gut, like signals from the stomach to cause evacuation of colon, that is the gastrocolic reflex, signals from colon and small gut to inhibit stomach motility and stomach secretion, the enterogastric reflex, reflexes from colon to inhibit emptying of ileal contents into colon, the colono-ileal reflex. Reflexes from the stomach and duodenum to brainstem and back to stomach by way of the vagus nerve to control gastric, motor, and secretory activity. Pain reflexes that cause general inhibition of the entire GIT, defecation reflexes that travel from the colon and rectum to spinal cord, and abdominal contractions required for defecation. Hormonal control, the hormones are released into the portal circulation and they exert the physiological action on the target cells with specific receptors for the hormone. So the principle for working of the hormone is the same as you have done in the endocrinology. The effect of hormone persists even after all nervous connections between the site of release and site of action have been severed. So it is very important table. You should all be learning about it. That Gastrin is the hormone, stimulus is protein, distension, and nerve. Site is the G cells of ventrum, duodenum, and jejunum, and action is to stimulate the gut function, gastric acid secretion, mucosal growth. Cholecystokinin function is to stimulate, released in the as a result of stimulus like protein, fat, acid, and the site of secretion is the eye cells of the duodenum, jejunum, and ileum, and the pancreatic enzyme secretion, pancreatic bicarbonate secretion, gallbladder contraction, growth of exocrine pancreas, inhibits gastric emptying. Then we have GIP, release as a result of protein, fat, carbohydrate, release from K cells of the duodenum and jejunum, stimulates insulin release, inhibits gastric acid secretion. Motilin, released as a result of fat, acid, or nerve stimulation from the M cells of duodenum and jejunum, stimulates the gastric motility, intestinal motility. Now, what is gastrin? Gastrin is secreted by the G cells of the antrum of the stomach in response to stimuli associated with ingestion of a meal, such as distension of stomach, the products of proteins, get the GIP, which is released by the nerves of gastric mucosa during vagal stimulation. So, the primary functions, actions of gastrin are stimulation of gastric acid secretion, stimulation of growth of gastric mucosa. Cholecystokinin is secreted by eye cells in the mucosa of duodenum and jejunum, mainly in response to digestive products of fat, fatty acids, and monoglycerides. This hormone strongly contracts the gallbladder, expelling bile into small gut, while the bile in turn is going to play important role in fat emulsification and allowing the digestion and absorption of fat. Also, it is going to inhibit the stomach contraction. Therefore, at the same time, this hormone causes emptying of the gallbladder, and it also slows the emptying of food from stomach to give adequate time for digestion of the fats in the upper intestinal tract. Now, cholecystokinin inhibits the appetite to prevent overeating during meals by stimulating the afferent nerve fibers in the duodenum. These fibers in turn send signals by way of the vagus nerve to inhibit feeding centers in the brain. Then we do have secretin, which is the first GIT hormone to be discovered and secreted by S cells in the duodenal mucosa in response to acidic gastric juice emptying into duodenum. Secretin has a mild effect on the motility of GIT, and it acts to promote pancreatic secretion of bicarbonate, and in turn is going to help to neutralize the acid in the small intestine. GIP is secreted by the mucosa of upper small intestine mainly in response to fatty acids and amino acids, but to a lesser extent in response to carbohydrate. It has a mild effect in decreasing motor activity of the stomach and therefore slows emptying of gastric contents into duodenum when the upper small intestine is already overloaded with food products. GIP at blood levels even lower than those needed to inhibit gastric motility also stimulates the insulin secretion, and for this reason is also known as the glucose dependent insulinotropic peptide. Motilin is secreted by stomach and upper duodenum during fasting, and the only known function of this hormone is to increase GIT motility. Motilin is released cyclically and stimulates waves of GIT motility called interdigestive myoelectric complexes that move through stomach and small gut every 90 minutes in a fasted person. Motilin secretion is inhibited after ingestion by mechanisms that are not fully understood as yet. So, this is all about the today's lecture. I hope you'll all get through it, through this, and do a good reading from your book that is Titan. And inshallah, definitely It'll be very much clear to you all after going through the lecture and from the book, respectively. Till then, stay blessed. Allah Hafiz.